Hello everyone. So before resuming the session, I would like to request the convener, Professor Sarath Delai, or both the coordinators, Aarti ma'am and Bhagwati ma'am, as well as all the speakers to assemble on the stage for a group photograph. Okay, so now that we have had lunch, let's resume with our previous session. Arvind sir is already here to resume back as moderator. And the next talk is by Dr. Amit Awasthi, so sir will be introducing him in detail. Thank you.
Welcome back, everyone. And after such a nice lunch, I hope Amit will be able to keep you awake. So that responsibility is yours, Amit, to keep everybody awake and listen. We are certainly enjoying, and I hope you are enjoying the talks as well. Do ask questions, right? So about Amit. Amit, I know since I joined, after NCCS I joined, Amit joined, he was a student in NCCS under Bhaskar Saha, one of our colleagues, wonderful immunologist. He got training, then as usual he went abroad, did his postdoc, came back and joined THSTI, and he joined early days actually when THSTI was not in the cluster. So now both RCB from where I come from now and THSTI both are located in the Biotech cluster in Faridabad. This was not there, and Amit joined much before that. He is doing fantastic work, and we are really very proud, particularly because he is also a student of his work, right? And doing and is very well known to the immunology community, not only India, but throughout the globe. And he has people praising him for his work, right? And recently we had a conference where he invited and everybody was talking about him and his work. They also feel so nice looking at him is growing. So with that introduction, I'll request Amit to start. He's going to talk on T-cell polarization, and as I promised you, I'll come back and tell you what complement does in T-cell polarization.
I hope I'll keep you awake after a nice lunch. And it is a privilege. This is my third time coming to this campus. And every time I come, I really enjoy this campus and the city. In fact, yesterday I was coming with Dr. Sahu, and the moment we get down at the airport, I said, looks very different than Delhi, because Delhi is more like chaotic, at least to me, but maybe at least to me. So I really enjoy here, and today I slightly change my talk title. Initially, I was thinking to give some data-driven talk, because I have sent an abstract for which I asked Dr. Dalai why we need abstract, because I have to give some basic things. Then I say, you start with basic, but give abstract. So I send my kind of abstract on Th9 cells. But then after seeing the talks, then I say maybe it's not a good idea to give data-driven talk. So I decided to give this a long widening road of Th17 cell differentiation. And as you know that previous speakers has really set up the stage, and especially thanks to Dr. Arora, who gave this nice antigen presentation, described how peptide fits into MHC class 1 or class 2, and that's how this whole thing started. So that thing I'm just leaving that side, and now I'm just imagining that the peptide is already fit into the class 1 or class 2, and then now it is being presented to T cell. So now I'll take T cell activation and differentiation. And some of the kind of not new subset, it started in 2006, now it's not too new like Th17 cells. I'll give you a story how it took longer to identify Th17 cells after 1984-85, when they described Th1, Th2 paradigm by Bob Kaufman and Tim Mossman. So for the students, when naive T cells, They comes out from the thymus after this positive negative selection. They comes to periphery in the lymphoid organs, and there they are actually sitting to see the antigen. Once the right antigen comes in, and these T cell gets activated with the two signals, CD3, I mean the TCR signal and the co-stimulatory signal, and then the third signal comes from the cytokines, and in this case, if the T cells hit with the cytokine called interferon gamma or IL-12, they become Th1 cells, and Th1 cells having a transcription factor which actually drive this whole program, and they produce interferon gamma, are important for elimination of intercellular pathogens like Mycobacterium, Salmonella, and so on. On the other hand, the Th2 cells can be generated from the same precursors with the IL-4, and they have a different sets of cytokines are being produced. They have different transcription factor, and they are important for clearing extracellular pathogens by activating mast cells, and that's how they but once these cells get dysregulated and identify the self antigen, they can be they induce autoimmune diseases. In this case, Th1 cells, they induce organ specific autoimmune diseases, and in this case the allergy and asthma. So these are the two subsets, and then in 2006 the differentiation factor of Th17 cells were identified. In this case, TJ-beta and IL-6, when you put together two cytokines, it induces a different program than Th1 and Th2, and the transcription factor drives this whole program is ROR gamma T, and that's what we call it master transcription factor of Th17 cells. The Th17 cells produce these cytokines IL-17A, F, and IL-22. And these cells are important for clearing extracellular pathogen, particularly fungal pathogens. The people who is having mutation in the Th17 pathway, particularly STAT3, these individuals are not able to clear fungal infection, and as a result, they have a lot of fungal accumulation. Nonetheless, I mean, these cells were now, described more in terms of autoimmune diseases than the fungal infection, and every organ specific autoimmune disease is now being associated with the Th17 cells than the Th1 cells. And this is a time when the T helper cell subset being like new subset were being discovered, and this is the same time we also identify in the same lab that two cytokines makes a new program of Th9 cells, and these Th9 cells make IL-9, and they have different transcription factor than all these different cells, T helper cells, and they induces different functions. And this is what my lab does a lot of work on the Th9 and Th17 cells. So we know these are the effector cells which are important for effector functions. On the other hand, we have a regulator, and Vinita briefly actually talked about the regulatory T cells, and these regulatory T cells actually primarily the major regulatory subset is the FOXP3 positive Treg cells. These Treg cells are generated in the thymus, but nonetheless, the FOXP3 positive Treg cells can also be generated in the periphery, and we call these cells are induced Treg cells. Both are expressing the same transcription factor, both are suppressive in nature, both controls. However, the stability of natural occurring Tregs and the iTregs are kind of differ. Some are more stable. That means the natural occurring Treg cells are more stable than the induced Treg cells. The other subset of regulatory T cells were this is after the discovery of Th1 and Th2 cells, when the Th17 and Th9 were not there. So people thought they identify some subset of T helper cells which could be a regulatory nature, and they call it T helper 3 cells, which over time is not really stand out. So the other subset of regulatory T cells, we call them type 1 regulatory T cells. This is what we identify that these cells are actually produces copious amount of interleukin 10, and interleukin 10 is one of the anti-inflammatory cytokines which actually not only control T cell proliferation but also actually suppresses antigen presentation. So that's how the IL-10 producing TR1 cells, which is FOXP3 negative, are also important regulatory T cells which play a major role, and we have identified how these cells are generated. So instead of IL-10, if you put IL-27, which is one of the cytokines, when IL-27 hits the naive T cells, they convert into IL-10 producing TR1 cells. So now I just quickly go back that you need two signals for T cell activation. obviously is the TCR with the right MSC peptide, and then you need a co-stimulatory molecule which is CD28 expressing on T cells, and the B7 family molecule CD80 or 86 on the antigen presenting cells. So in this case, when you have the two signal, you have the cytokine production, T cell proliferation, and effector functions. But you have certain condition in which this two signal is not actually there. You have TCR signal, but the second signal is somehow abrogated, and there are examples, for example, HIV or infection condition like, for example, leishmania and other infection, where the second signal is kind of down regulated, and as a result you have this signal is less. As a result, the T cell is actually not proliferating, and this is stage called T cell energy. So, so T cell differentiation is after T cell activation. So T cell need to be activated, and these activated T cells actually producing IL-2. IL-2 is a growth factor which actually make these T cell proliferate. And during this, at the same time, you have other cytokines are being produced which actually drive T helper cell differentiation in different pathway, Th1, Th2, Th17, and so on. So this is how it started. This is a classical paper of JI, what these two gentlemen has published. And I briefly mentioned that this is either Th1 or Th2, and these subsets are actually mutually exclusive. So basically, if you have more Th1, the Th2 subset is suppressed. What Vinita actually mentioned, this could be a one possible way of identifying potential therapeutics. And if you have more Th2, the Th1 subset actually are suppressed. In fact, people have done the experiment that other than the cytokines, if you actually take the master transcription factor of Th1, which is TBAT, or the master transcription factor of Th2, which is GATA3, if you overexpress TBAT in Th2 cells, it shut down the Th2 program and convert these cells into Th1, and in a vice versa also. So, this is how they started. This is the original paper. This is actually some of the paper. Journal of Immunology actually republished these paper as a pillar of immunology, and this got republished in 2005, and this is what the original data that they have made different Th1 and Th2 clone, and Th1 clone is making interferon gamma, and Th2 clone were making IL-4, and this is what. And each of these clones at that time were suggested that they will have function in helping B cells. So the T helper cells mean helping other cells, particularly B cells. So that's what the name of T helper cells. So in case of Th1, they make interferon gamma, and these plasma cells make different sets of antibodies. And in case of Th2, it helps plasma cells to produce different sort of isotype of these antibodies. So how this is started? So you have these pathogens, as Dr. Arora actually mentioned about the PAMs and the PRR, and these pathogens actually come in contact with the antigen presenting cells. I just depicting it dendritic cells, but it could be macrophage as well. So these pathogens, when it senses on the cell surface, it senses these toll-like receptors. And this sensing of toll-like receptor make these cells become more activated. So this sensing is making the cells activated. As a result, the phagocytosis actually increased. And after this phagocytose, these pathogens, they convert into, they chopped into small pieces through the endosome and lysosome, and then they come into the MSC molecule. And during this sensing, the sensing makes these cells to produce pro-inflammatory cytokines. In this case, I'm actually mentioning this IL-12 is being produced, and this IL-12 goes and binds to its receptor on the T cells through IL-12 receptor, and this signaling lead to the activation of T cell, and that produces interferon gamma, and this interferon gamma actually comes back in an autocrine manner to increase the Th1 cell differentiation. So here is the initiation, and here is the kind of amplification in case of Th1 cells. So let's go a little bit deeper into the biology of Th1 cells. I'm not really touching Th2 cells too much, because the Th17 cells discovery somewhere lies into the go deeper into the Th1 pathway. So if you see this IL-12 receptor on Th1 differentiation, IL-12 cytokine is a heterodimeric cytokine, is composed of P35 and P40. P35, P40 is nothing. It's like when you run this protein, it comes at the 35 kilodalton and 40 kilodalton. That's what they call it, P35 and P40. When they combine, they make P70, which is a active form of IL-12. And this active form of IL-12 actually bind to its heterodimeric receptor, which is composed of IL-12 receptor beta 1 and IL-12 receptor beta 2. This complex actually activate the cell signaling, and this cell signaling happens through the STAT4, which is a kind of signal transducer in case of this IL-12 signaling, and that actually lead to the Th1 induced Th1 response. So each of these processes has been shown in a kind of system where the mice were kind of made deficient. Each of these pathways like IL-35, P40, if you take these knockout mice and if you actually inject mycobacterium in this case, these mice actually are susceptible and they are not able to control the infection, and because they don't have Th1 response. And similarly, if you actually take the IL-12 receptor beta 1 or beta 2 mice, they have the same phenotype like IL-12. They are kind of not able to control the infection, and they have no Th1 response. And similarly, you have STAT4 knockout that also does the same thing. So this is how the immunologists actually prove that each of these pathways are important in vivo to make Th1 cells. And this is the same thing like I told you, interferon gamma. Interferon gamma is also composed of this heterodimer, and that actually induces JAK1, JAK2 signaling that lead to STAT1, and that induces Th1, enhanced Th1 response. And this is again done the same way that if you make either of these knockouts, you have the same issue that the infection is uncontrolled and you have lesser Th1 cells. So these observations were also kind of correlated when you have the individuals or human being who is having defect in interferon gamma signaling, or they have a mutation. They cannot really control the infection and they have much lesser Th1 cells. So now I just briefly mentioned to you, this is the one pathway which initiates the Th1 differentiation, and it goes all the way, induce interferon gamma comes back and induces STAT1, and that's how this whole amplification and gene program of Th1 cells actually being induced in this case. But nonetheless, then this was actually known, and then I think this is a time in 2000, Laurie Glimcher group was actually trying to identify that master transcription factor that actually drives the Th1 pathway. So what they have done, they have taken these naive T cells, they did all this kind of screening, and what they found, actually they made the clone, and what they found that one of the transcription factor actually being expressed specifically in the Th1 cells at different time points. So if you take these Th1 or Th2 clones at different time points, you see TBAT, which is a TBX21 gene name, is expressed only in the Th1 cells, not in the Th2 cells. And then they try to identify the interferon gamma, they screen the IFN gamma locus, and they found the TBX21 binding site in the IFN gamma promoter. And then what they have done, they did the luciferase assay, where actually they put this 3 different luciferase. One is the IL-2 luciferase, IL-4 luciferase, and interferon gamma luciferase. And then they co-express this TBAT in the cell where you have this luciferase in place. And what they actually could see that you have, when you put the TBAT, you have more interferon gamma, but there is nothing is happening in case of IL-4 and IL-2. And that's what they also did, this retrovirus expression of TBAT in the T cells. And you see at any time point, if you overexpress this TBAT in the T cells, in the primary T cells through the retroviral expression, you have more interferon gamma. So that's how they published this paper in Cell. And that's how they come to this model where you have this initiation of STAT4 goes to TBAT, and this STAT1 again goes to TBAT. So that's how the TBAT is a master transcription factor of Th1 cells. So far I just mentioned to you that IL-12 and interferon gamma induce T cell differentiation to Th1 pathway. Transcription factor STAT1, STAT4 are essential for development of Th1 pathway. TBAT is found to be a master regulator of Th1 cell, Th1, and Th1 cells are crucial for cell-mediated immune response against intracellular pathogens. So this was the time when we were only knowing about Th1 and Th2 cells. We were not knowing about any other subset of T helper cells. So everybody was trying to put their model or their disease either fit into Th1 pathway or Th2 pathway, especially the T cell biologist. So this time, what was happening that, people who are coming with the multiple sclerosis or rheumatoid arthritis and other autoimmune diseases, these clinicians were trying to see what kind of cytokines they have in the lesions of these individuals, and they found that interferon gamma and Th1 cells are kind of being accumulated in the lesions of the MS patient, and then reactivation of these cells make interferon gamma. And then they also did some mouse experiment when they make this Th1 clone outside, and they adaptively transferred these clone into the mice. These mice actually have a sign of EAE, which is a kind of mouse model of multiple sclerosis. And similarly, when they make this myelin-specific Th1 cells transferred into a mice, these mice also generate this EAE signs. So I just want to tell you what is EAE. It's an experimental autoimmune encephalomyelitis, a mouse model for multiple sclerosis. This model has been used by immunologists, particularly T cell biologists, or to really address T cell pathway because for multiple reasons. One is that you know the peptide very well. This is a well characterized peptide. You know where it fits. It's a class 2 peptide. So that's how you can actually identify and study the T helper cells in this case. So what this model looks like, so what you have to do, you take myelin antigen. Myelin antigen could be many. It could be myelin basic protein. It could be MOG peptide and PLP. And if you immunize the mice, they get kind of paralysis, and then you can study the T cell activation in this case. And this is how the scoring is done in this case. If you immunize mice at day 0, around day 10, you start seeing the sign in the mice. They have the limp tail, they have gating problem, and then they ultimately get paralysis. And then you can clearly take out the brain or the CNS from these animals, and you can see a kind of distinct lesions in the brain or CNS of these animals, which is having EAE. And this is how the mouse looks like. You have to really feed them because they cannot really, they're paralyzed, and you have to give the gel food in the cage so that they survive. So this is something which this was really long time the immunologists really putting this under the carpet, and they were not really talking about it. So what they were doing, they have taken all these molecules, all these mice, which were Th1 associated molecules, and they immunize these animals, and they start seeing the phenotype. And if you see the two animals in this case, which is IL-12P40 and IL-12 receptor beta 1, they have no paralysis, and they have no Th1 response. So this is something, a paradox, we call it paradox, and this was long time was kind of unexplained until these couple of paper came out. One paper came out, Immunity, in 2000, that they have identified a novel P19 protein engages P40 and this cytokine called IL-23. And then they also identify a receptor for IL-23 subsequently in 2002 in Journal of Immunology. And now it looks like, so you know this is IL-12, P35, P40, IL-12 receptor. But then you have new subunit of the cytokine which takes P40 with its as a partner, and then you have a receptor of IL-23, which is IL-20 receptor alpha, takes IL-12 receptor beta 1. So now it's kind of very complex biology of IL-12 and IL-23. And this was perhaps one reason why the Th17 cells took so long to be discovered after Th1 and Th2 cells. So again, now if you really see here in this case, if this mouse having no IL-12, but this mouse also don't have IL-23 because this is a sharing subunit. In this case, IL-12 receptor beta 1 having no Th1 cells, but they don't have also Th17 cells because this receptor is also shared with IL-23. So this is the classical experiment which actually solved the problem by Dan Koa in Nature, published in 2003. What actually Dan Koa did, he has taken 3 different knockouts, P35, P40, and P19. And then, so this we know, this IL-12, this we know IL-23, and he did simple immunization of these 3 different knockout, and then did the EAE experiment where he just simply see the phenotype of EAE. So P35, paralysis. P40, no paralysis. P19, no paralysis. Then he opened up these mice and isolate the T cells from these mice, and what you could see, there is no Th1 cells, but it's still paralysis. They have more Th17 cells. In this case, no Th1 and Th17 because IL-12 is not there and IL-23 is also not there. And in this case, Th1 cells are there, no paralysis, but you have no Th17 cells. Here is P19. So this is actually suggesting that it is IL-23 mediated Th17 cells that is actually essential for induction of autoimmune diseases. It is not really IL-12 which was doing the job. So previous identification of Th1 with the lesions of MS or in autoimmunity model was kind of a correlation. It was not really proven genetically, and this experiment clearly indicates it's not Th1, it is Th17 cells which induces organ-specific autoimmune diseases. And same thing experiment has been done with the IL-12 receptor knockout. So in this case also, if you don't have beta-2, no Th1, still paralysis. If you don't have beta-1, no paralysis. There is no Th17 cells. And here if you just take out this, in fact we made this mouse where we remove IL-23 receptor, and then you see these mice are resistant to AE and they don't have Th17 cells. So both through IL-12 or IL-12 receptor or IL-20 receptor, it is clearly indicated this is the cell is causing the thing. So the time of this Th17 and T cell differentiation, this was very exciting time for the immunologist. And this is the time the subset of FOXP3 was also identified around 2001. But the original discovery of regulatory T cells was done by Simon Sakaguchi when he was doing a postdoc. In 1995, he published this classical paper, which is cited so many times. And what he was trying to say that a small subset of CD4 cells in the mouse is always having CD25 on its surface, right? So if I just briefly take you back, CD25 is an activation marker of T cells. So anytime T cells get activated, you have CD25 expression on the surface. But nonetheless, what he was trying to say that mouse without any manipulation or any immunization, still you have subset of T cell which is expressing CD25. So he called these cells regulatory T cells, and then he went back and see from where these cells are generated. Then he did this thymectomy experiment where he removed the thymus in the neonatal mice, and these thymectomy mice later on become autoimmune because the regulatory T cells which are generated in the thymus are not there in the periphery. Subsequently, after 1995, it took like 2003, the three paper came, two paper back to back in Nature Immunology. One was in Science by Sakaguchi himself. Came up and say whatever the cells he has identified is having a now transcription factor. Remember, so anything we talked about immunology, particularly in T cell, when we talk about this new subset, we always want to have a kind of what is the transcription factor of that subset. For example, Th17 I mentioned root gamma T, Th1 I said Tbet, but in this case they say it's a FOXP3. And then a lot of work has been done by Sakaguchi and Sasa Rudensky, who really describe this phenotype and phenomena in multiple ways. If you remove FOXP3 using the scurfy mice, where the natural mutation in the FOXP3 locus, these mice actually die very young age. If you have the individuals who's having natural mutation in the FOXP3 gene, we call them IPEX syndrome. And these individuals also really die because of multiple organ failure, because you have more other effector cells goes into the different organs and do the damage of the organs. I don't know how much time do I have. So this is again, so now we talked about a little bit about inflammatory T cells, Th17 cells and Th1 cells, little bit about regulatory T cells. And this is the two paper which I think is very important to describe to the student that how the regulatory T cell. Now people are talking about can we do adoptive transfer therapy of regulatory T cell. Let's say if I have autoimmune disease, you take out my T cells, you somehow make them regulatory in nature by putting cytokine or maybe do some sort of, make them FOXP3, put those cells back into me. Now the question is whether those cells remain stable or not, right? So in this case, the two paper, one was from the Ruslan Madjitov's group, what he has suggested, that if you co-culture the regulatory T cells with the effector T cells, that means non-FOXP3 cells, the regulatory T cells able to suppress the T cell proliferation. But if you put some pro-inflammatory cytokine into it, which is interleukin 6, now regulatory T cells are not able to suppress. The similar experiment we had done when I was doing postdoc in this Nature Medicine, what we published, what we found that if you take out the effector T cells from the mouse which is having inflammation, you take out the T cells from the brain and you try to put these T cells with the regulatory T cells, is not able to suppress because of the inflammatory nature of these T cells. But if you do the same experiment, if you put these cells with the regulatory T cells, if you put anti-IL-6 antibody, these regulatory T cells start functioning and the T cell proliferation is kind of suppressed. So this is a kind of original figure from the paper, and this is, I just make a depiction just to make you understand kind of easily. So Treg cells are non-functional in the inflammatory environment. This is what people are talking, and that's why the regulatory T cell therapy is still not reaches to the level where we should have been. Now I know the people working on making this stable Treg cells, especially the company in the Seattle, and I think it happened that this guy was actually came to a meeting which we organized last week, where he actually making a way to make this regulatory T cells very stable by infecting these T cells with the right FOXP3 clone. So now, I mean, so far we were knowing the Th17 cells through the IL-23 perspective, through the IL-23 receptor and IL-23 cytokines. But still the differentiation of Th17 cells were not known. Until in 2006, there were 3 groups actually working independently. This paper came out from the MRC London, from Brigitte Stockinger. What she was trying to do, she was trying to do an experiment where she was co-culturing 3 different cell types in a Petri plate, is a naive T cells, regulatory T cells, and the dendritic cells. And they're trying to see if you go culture these cells, what happens, which direction they go. They become Th1, Th2, or different cell types. In this case, they found there is no Th1, no Th2, no Th17 cells. And then what they have done, they put these 3 conditions and they put LPS also in together with this. And they found that it inducing a lot of Th17 cell differentiation in this case. And then what they smartly did, they removed dendritic cells. With the IL-6, we know LPS induces IL-6 through the dendritic cells. So they removed dendritic cells. With the IL-6, they removed Tregs with the TJ-beta, because Tregs also produce TJ-beta, and TJ-beta is also not only secreted by the Treg cells, it is also on the surface in a form of lap. Right? So then they replace these 2 cells with the 2 factors, and that's how they identify IL-6 and TJ-beta 2 together, making Th17 cell differentiation. So this was the Brigitte Stockinger published 1 month before Vijay Kuchru's group, where he was trying to do a different way of doing experiment. What he was trying to do, he made a mouse of TJ-beta transgenic mouse under the CD2 promoter. So every time your T cells are activated, it will secrete TJ-beta, right? So they were trying to do an experiment that if you immunize these mice, and they were expecting this result because they were thinking TJ-beta is a suppressive cytokine. So they were saying this result should be like this. But every time they were getting a result opposite. So this was the actual result. They were saying every time a paralysis. So every time the postdoc used to go, that this is the result, he said repeated, because that's not they were actually expecting. And then a separate person was doing another experiment where they have made one mouse, which is FOXP3 GFP knock-in mouse. Briefly, I mentioned to you, FOXP3 is a master transcription factor of regulatory T cells. So what they were trying to do, can we make a mouse in which the FOXP3 is tagged with the GFP so that we can see the green versus non-green cells in the mouse in the in vivo conditions. So they made this mouse, and then they tried to identify how we can make non-Treg cells into Tregs. So obviously they were making TJ-beta induced Tregs, we call them iTregs, and then they were also trying to say what are the cytokine can suppress this TJ-beta induced Tregs. So therefore, they tried to took all the cytokine, whatever known at that time, and they started doing experiment that you have the TJ beta alone induces a lot of FOXP3, but when you start putting these cytokine together with TJ beta, you see some of the cytokine is suppressing this FOXP3 expression, and one of the cytokine was stand out is interleukin 6. And then we took this supernatant from this culture supernatant, and they measured different cytokine using Luminex, and they found that IL-17 is found to be induced in this particular condition. That's how they independently identify that 2-factor is important for, and this is the original experiment. When you have the TJ beta, you have FOXP3 induction, and when you have TJ beta plus IL-6, the FOXP3 is suppressed, and you have this IL-17 is coming on this side. So then subsequently they identify IL-21. I'll just skip this, and then they, based on this, at the time I was there, so I could also contribute to this. So what we propose that, naive T cells have different way of going to Th17 pathway. One is the differentiation stage where you have the 2 cytokine make this T cells to be start programming through Th17 cells, and during this process IL-21 comes in, and this IL-21 amplify this developing Th17 cells, and that's what we call amplification. And then during this amplification you have IL-23, which otherwise don't express on the naive T cells. And that's what the IL-23 is actually make this stabilization of Th17 cells. So far, I have still time. So I just skipped that. So thank God, the data slides are now. So I don't want to go with the data slide. So what we can say that after Th17 cells, all these different organ-specific autoimmune diseases are now associated with the Th17 cells, and this is already proven in the mouse using IL-17 knockout or IL-23 receptor knockout or anti-IL-17 neutralizing antibody, that these are driven by the Th17 cells. And in fact, psoriasis is one of the example where secukinumab is anti-IL-17 antibody. It was approved by the FDA, and this antibody actually does wonder to the psoriatic patient. If you inject this, you infuse this antibody, the lesions of psoriasis lesion goes down significantly. And now the second antibody coming in the market by Janssen & Johnson is anti-P19, which is IL-23 receptor, IL-23 cytokine antibody, which is even much better than in this case, the PSI score is around 70, but in case of P19 or IL-23 cytokine antibody, it is coming around PSI 90. So that's even much better for this. I think with this, I will stop here, and then still we are working on this pathway, and we have identified the prominent drug target using ROR gamma T, which I was thinking to show, but now I don't have time. Thank you so much.
Amit, so what happens to FOXP3, which is already present in these cells, when basically they lose out the expression? So it undergoes degradation, or I can understand that new FOXP3 is made. What happens to this?
So you mean to say what happens to FOXP3?
Which is already transcribed and translated, and these cells are expressing that.
So in case of naturally occurring Treg cells, the FOXP3 expression is not normally loose. Actually, they contain it. But the problem is in an inflammatory condition, although they have FOXP3 expression, but these T cells, which is effector T cells, they cannot really suppress, because when you have this inflammatory milieu, the T cells start proliferating faster than this FOXP3 able to control in case of natural occurring Tregs. But the induced Tregs, people have done the experiment. If you do induce Tregs with the TJ beta and put them in the rag knockout mice, and over time they lose FOXP3 expression and they become effector cells. They start making interferon gamma and other cytokines. So there's kind of difference between non-Tregs and iTregs.
Yeah, this is what I'm asking. So what happens to already expressed FOXP3? So it also remains in these cells, or?
So what happens in case of when you put iTregs into a rag knockout, and rag knockout is kind of empty. So when actually the T cells goes into empty environment, they start proliferating homeostatic proliferation. And because of homeostatic proliferation, the new cells budding out don't have FOXP3. Yeah, that way.
Thank you for the very good presentation. So just one question was there that we keep reading about the dual effect of IF and gamma. So during the initial inflammation, it plays a role in inflammation, and subsequently it also has anti-inflammatory role. So could your mix of T cells with Tregs did not function because of high levels of IF and gamma and not the IL-6 story?
So the paper got published in PNS about interferon gamma, but it is not, if you put interferon gamma together with this Treg co-culture, it is suppressing the function of Treg, but not the level of IL-6. Now the second thing is what people are talking about interferon gamma is in the kind of pro-inflammatory making Th1 cells. But interferon gamma is actually, the new literature start coming out that it is having anti-inflammatory property as well. So initially, it may be particularly for the infectious diseases is important. It's very important. Th17 is not really important for intercellular pathogens, but interferon gamma is very important for intercellular pathogens. So, for example, take the example of Mycobacterium tuberculosis. If you have interferon gamma, it's going to clear the pathogen. But if you have interferon gamma, it also what it actually doing, and some paper from NIH is showing that this interferon gamma make T cells to make also IL-10. So this kind of, that actually is a kind of negative feedback loop. So if you have more interferon gamma, more interleukin 10, anti-inflammatory pathways start kicking in. So it's kind of, it is having both role.
Yeah, here,
Yes,
one.
sir. Yes, sir. Sir, the thymectomy of mice that was done, so that would downregulate all the T cells. So how are you particularly checking for T-Rex cells?
So for example, before the thymectomy, whatever the T cells came into the periphery, they are still there, right? So new things are not coming. So, and particularly this, the Treg cells, which is generated in the thymus, is not going to be out because they are all generated in the thymus. And as a result, if you keep these mice longer, then this absence of regulatory T cells will make these mice more susceptible to have this autoimmune kind of symptom like, diabetes and all those kind of other stuff.
Sir, but also in the periphery, like if we before we remove, there were already the T cells which were present in the periphery. So there maybe there is also T-Rex cell in that. So how do you like already
That's a
present?
very good question, but the Tregs always in a much lesser than the effector cells. If you really see the kind of ratio, so normally it's only 10% of total CD4 in case of mouse, right? So that ratio is kind of dysregulated.
So one general question. So that he induced that mice model shows the paralysis and the immune role of the immune systems. So this paralysis has anything to do with the many other kind of disease also cause paralysis, and the similar kind of immunological mechanism or pathways involved, or like stroke also has paralysis and many other.
So you have, this paralysis like you are mentioning, but in this case we were knowing very well that, the what kind of, antigen we used and where it is targeting. So that's what this paralysis is not due to other complication. It's just because the T cells, which is now primed in the periphery going to the brain and chewing out this, neurons or the axons, and that's what the kind of, nervous system is kind of dysregulated, and that's the reason.
Okay, and for the autoimmune disease you mentioned, which is mostly people say is a very inflammatory disease. So just curious, you showed a bunch of cytokines profiling over here. Is there any way cytokine resistance happens? Like we have the example insulin resistance. Insulin is there, but it cannot get inside due to the signaling defects. Similar kind of cytokine signaling defect happens and resistance occurs over there.
I haven't heard cytokine resistance. Uh, you can say in a way that if T cells are hyperactivated, it can have two fates. One, obviously, it goes to apoptosis, and that is perhaps through the hyperactivation of antigen receptor. That is the case for that. But the the other thing is it can actually start expressing negative molecules, what Professor Arora mentioned about the negative checkpoint molecules. So they will start coming on the surface, and as a result, the T cells will start getting down in terms of proliferation and cytokine production.
And then one general question, immunologists always love to show everything is immunology perspective, but in some cases, the early phase of the disease when it's initiated, where is the glitch, why the immune cell fails and do not
No, see, immunologist doesn't want to show everything in the case of, but we use the right model system. You know, this is, see, when I use the EAE, why I use the EAE mouse? I could have used any mouse where I don't know even the antigen, for example, right?
But MS also happens without the infection.
No, MSC,
In general, I'm saying that.
as a T-cell biologist, I am using the mouse model, which I can say this is important.
Correct. That's
But
model.
MS is a kind of, if you see the full spectrum of MS, it is having microglia, astrocyte, many other
Myelinations,
things, reactive oxygens, nitric oxide. People has done the experiment in NOS to knockout and all those. So, but when you have to identify something, you have to first pinpoint one person, then Arora sir, then you. I cannot really jump on everybody, na? So I have to one after others. To
The democracy.
yeah.
I have one fundamental question actually. This is where I sometimes struggle in the classroom, maybe to you and also Vinita, madam. Because the signal 1 minus signal 2, well, let's say it induces tolerance. Are we talking this in the context of naive T cells or antigen experienced T cells? Because we know we have to give signal 1 and signal 2 to activate naive T cells. If you do not give signal 2, we don't activate them. On the other hand, antigen experienced cells, they also need some co-stimulatory molecules, signal 2. If you do not them, they stop functioning. So this is where many times, we get confused, what we are trying to say. One case, we are not providing activation signal. Another case, we are actually T cells which have already antigen experience, we are inactivating them. Can it be same?
I think she will be the right person to address. And I can comment, okay. So see, I think, the signal 2, what we talked about is a called danger signal, okay? That primarily comes from your toll-like receptor and other things. In normal scenario, our interaction, MSC and your naive T cells interaction happens continuously, right? But you need a danger signal that comes from the activation of innate pathway. That actually makes signal 2. I don't think I address your question. You are trying to say, I forgot. No, Vinita has to help me.
Normal case when we call T cells coming in contact with MSC, that is through self-peptide and MSC. In fact, that interaction is also required. But T cells will get specific signal when you have, non-self peptides. So in this context, we are telling that, let's say this is in vitro experiment we do. We take transgenic naive T cells and we do not provide any signal to, we don't activate them.
Not at all.
But the same cells, now we provide signal to can get activated. When we need to talk about tolerance, that means we have already made them inactive. So this is where, we have to find a distinction.
Only 5 minutes.
So to answer that question, it's not just one signal one. So we are talking about a primary signal, which is MHC peptide complex. Second signal is, say, cell-associated adhesion molecules like CD28 and B7 family that he talked about. There are additional signals which are also soluble signals in the form of danger that he mentioned. So cytokines secreted by the APCs which are already activated. So when naive T cells get activated, combination of all three to different extent is required, and a part of that is to do with how much is the affinity between MHC peptide complex and the T cell receptor. It's always a range. So if you have a higher affinity interaction there, you can, even for a naive cell, make do with a little less of signal 2 plus 3. Okay, so then activation happens. If it is a low affinity, you certainly need the whole one. That's why we talk about dendritic cells having all sorts of entities which will provide good signal. So Post activation, T cells always require, same T cell, if we are talking about divided in divided fashion, that same T cell requires fewer signals than it needed at the naive state. And this again, when I say fewer, I know I'm making a generalization, but how much of interaction of the cell surface molecule is required, how much of cytokine is required, all of this is something that one has to remember. Or less. That is why it's not a tolerance if there is inflammation. So if you take, for example, already there are SARS-CoV-2. SARS-CoV-2 specific T cells are already generated by vaccination. There is an Omicron coming in and it has infected mucosal lining. So if epithelial cells infected with SARS-CoV-2, right? These are not professional antigen presenting cells, so they have only MHC class 1, and because there is inflammation, there is danger. So there are some signals. These epithelial cells can very easily present antigen to pre-activated, meaning pre-exposed memory T cells and kill and get killed in return because epithelial cells are infected. So what I'm trying to say is that we know much more signal, we know much less signal. That is why non-professional antigen presenting cells can be killed by memory T cells and effector cells, but non-professional antigen presenting cells cannot trigger naive T cells. So there is a distinction between second signal, third signal, how much, but for every individual cell, how much of this and how much of that, I don't have an answer.
Thank you. Thank you. Thank you.
Thank you so much for such a wonderful talk, Amit.
Thank you, sir.
Thank you so much, sir, for this insightful talk. I request Dr. Arvind to present the token of appreciation to Dr. Amit. We are also thankful to Dr. Arvind for chairing this session, and I request Dr. Ravikant to felicitate Dr. Arvind Sahu.
So before that, as I promised that I will add complement to the story, I would like to add what we were talking about, the third signal, right? So can complement be a third signal? This question was asked because both the antigen presenting cells as well as T cells make complement, synthesize complement. And when there is these two interaction is going on, complement activation is there and there is generation of effector molecules C3a and C5a. So the question is, can this really matter? Does it really matter in terms? The answer is yes. If you have complement activation, you get Th1 or Th17. If you do not have complement, this C3a and C5a, what you get is Tregs, okay? So there is a nice article, you want to read more about the mechanism. Complement a day keeps FOXP3 away. Read it and you will know more.
Thank you, sir.